And now it's my distinct pleasure to introduce Linda Birnbaum. Linda is the director of the National Institute of Environmental Health Sciences, one of the National Institutes of Health, and the National Toxicology Program, and she's been in that role since 2009. She's had an amazing career, but very relevant to being here, and she spent most of that bravely as a federal employee. Dr. Birnbaum has received many awards and recognitions, but for the sake of being here today, one of the most important is being elected to the Institute of Medicine of the National Academies, known as the National Academies of Medicine in 2010, which really is one of the highest honors of all of our fields. So it's a great pleasure to welcome you.
So I really want to say how happy I am to be here. As you know, many of our colleagues are not able to be here. They're not working. I have 12 people who run our Superfund program, and they are furloughed. But as well as you know, there were a total of about 800,000 federal workers, 4 million contractors, and untold numbers of cab drivers, luncheonette owners, coffee shop people who are not fully employed at the present. And I think it's a terrible thing that's going on. And worse, this is the worst one we've ever had. And I'll just tell you, for someone who lived through several shutdowns, including the 95-96 one that spanned Christmas and went for 21 days, it took years for federal employees to feel that they were valued and felt secure in their jobs. So I expect there'll be a lot of results or fallout from this. So now to switch more to the science and talk about the great stuff that I want to talk about today. Let's see, how do I do this? Oh, that's... Sorry.
Yep, no worries. Just push the green.
Oh, the green arrow. Sorry. Okay, so what I wanted to do just first, and this kind of actually gets to what Robert was talking about, is the need for interdisciplinary, multidisciplinary, transdisciplinary work. This is our new strategic plan, and I just came out a couple months ago. This is the whole one, so it's easy reading. You can easily download it, and I've put a bunch of flyers out there that kind of talk about the themes and the goals. But the point is everything goes together, whether it's, for example, the basic sciences, which is, for example, advancing environmental health sciences, whether it's promoting translation, actually making data to knowledge to action, or whether it's actually getting the word out, training people, increasing scientific literacy of the population and so on, all of those things, and they all have to work in an interactive way. So I just wanted to, I guess, put a little plug for that, for the kind of work. So you can understand why we have funded this efforts of EHESD for about, I think it's about 14 years actually, close to 15 that we've been funding this, and really does some exciting work, pushing the boundaries. I love Melissa's comment about it's a beacon. That's a nice image for the field of environmental health. So I personally have been pushing the intersection between infectious agents and environmental health for quite a long time. But I've been trying to convince some of my colleagues at NIH that exposure to environmental contaminants can alter the immune response to pathogens. But of course, the pathogens can alter the response to the toxicants as well. So we've got a two-way street that we have to look at. For some reason, I guess because it was very difficult to, for example, culture, say, gut microbes, because they're anaerobic in many cases. And the point is we ignored them. And now, of course, we're focusing not only on the gut microbiome, but all the, I think there were 22 microbiomes, 14 alone on the skin, and realizing that there are many more of them than there are of us. And we need to have a better understanding of how they can alter our response to environmental stressors, just like environmental stressors can alter their composition, for example, and what we have. And then, as Robert mentioned, we have the whole issue of climate change and ecosystem changes that are going on that can impact us. So when I define environment, and I think this is important to understand, what NIEHS talks about environment, we're not just talking about synthetic chemicals. We're not just talking pesticides. We're not just talking about chemicals and consumer products. We're also talking about food. Guess what? Nutrients are made up of chemicals. We're talking about, for example, our infectious agents. We're talking about the microbiome. We're talking about things. Somehow people forget that the drugs that you take are chemicals and may interact, for example, with our other environmental exposures. And, for example, there's growing interest in, for example, chemicals in consumer products and personal care products and so on. So I said I've been interested in this area for a long time, and I actually was a senior author on a paper that was published when I was at EPA. This is over 20 years ago, where I've kind of always been interested in the persistent kind of chemicals. So I'm still interested in the dioxins and the PCBs and the flame retardants, but I'm really interested now in the PFAS. But the point here that I want to make is that we were able to show that the most sensitive response that has still ever been reported as a single acute dose to TCDD was that it altered the influenza host viral response. So in fact, at a dose of 10 nanograms, single dose that you gave, the animals then died from viral infection instead of being able to recover. So it's a clear interaction between the toxicant exposure and the environmental trigger, in this case, dioxin. And I should remind you that this kind of response has now been shown in lots of animal species. It's not just mice, it's also rats, it's also non-human primates, and not just for something like dioxins or PCBs, but also, for example, and I'm going to show you some of the data for the PFAS, but it's also been shown for things like how air pollution can impact the response not only to influenza, but to, for example, strep infections or staph infections. So you have this interaction going on where host susceptibility is different and the impact of the viral or the bacterial infection changes in concert with the exposure that goes on. So Robert talked about One Health, which I'm a firm believer in. If we don't learn from the critters around us, whether they're domestic or wildlife, and even the plants, we are bound to ignore warning signs, for example. So in the late 80s, when many of you know the Baltic was heavily contaminated with a lot of persistent pollutants, there were die-offs of seals in the Baltic, and it was due to a virus related to the distemper virus of dogs. This has happened in other parts of the world since then, the same kind of thing, and it was shown to be associated with elevated levels of PCBs. Just recently, I'm talking about within the past 5 or 6 years, there have been major die-off of harbor seals off the coast of Maine, again, related to exposure to different kinds of persistent organic pollutants. And we know that now, as I said, dioxins, PCBs, and polychlorinated dibenzofurans were implicated as immunosuppressors, leading to the die-off. So it's an interaction, again, between environmental stressors and the infectious agents. So does this occur in humans? Well, the answer is yes. So one of the standard assays to look for an impact on the immune system is the suppression of the sheep red blood cell response. And that's been shown from, for example, PCBs and dioxins and PFAS in mice, rats, and non-human primates. And we see essentially an identical response occurring in children associated with perinatal exposure, both late pregnancy and early postnatal exposures of the mother, and this suppresses the response to vaccination. So I'm busy asking, every time I hear about a new measles outbreak or a whooping cough outbreak, is it really because it's due to the vaxxers who are not vaccinating their kids? Or is some of it due to that we have a percentage of our population who have not mounted a therapeutic immune response? And Philippe Grandjean and coworkers looking at the Faroe Islands, where you have the beauty, and I say that kind of every time I talk about exciting science, I say how wonderful it is, and yet it's really bad things that are happening to people. But here you have people, a very homogeneous population who are living basically in the North Sea. There's no production of PCBs, there's no use of PCBs there, but they are fishers and fishermen, and they also eat whale, and so they are heavily exposed to contaminants that bioaccumulate up the food chain there. And he has been following cohorts of children since the mid-1980s now. In fact, some of them are no longer children, but are 28, 30, 32 years old, and he continues to follow them for effects. But what he was able to show was with the PCBs that there was a decreased antibody response, and about 20% of the population was not mounting a therapeutic response to tetanus and diphtheria. Recently, he's done some studies with the PFAS chemicals, to which we are all exposed. Every single one of us has some of the PFAS, at least, in our bodies, and it's a worldwide issue. And they're finding, again, he's done a number of studies here. He looked at kids at age 5, age 7, now he's looked at age 13, and decreased antibody levels for tetanus and diphtheria. So I think that these are pretty important evidence of what is occurring in the human population. And I happen to be one of those people who do believe that when we have mechanistic insight, we have animal studies, and then we have observational human studies, I am pretty much believe that we have or are approaching causation in that case. So another point is air pollution. I already mentioned how air pollution, for example, both PM10, PM2.5, ozone, I think the NOx as well, can suppress the response to different kinds of experimental studies to flu and bacteria as well. And there's growing evidence that in household air pollution, especially in parts of the developing world where people are cooking over traditional three-stone stoves, burning different kinds of biofuels, the levels of air pollution in the huts are extremely high. They actually at times can make Beijing's air look relatively clean when you see what's going there. But there's actually evidence that these exposures are part of the reason that not only is there a very high rate of respiratory infections, both in the children and in their mothers especially, but actually Kirk Smith has shown us some work actually more recent than the one cited here from 2011, but that in fact, when you change out those stoves and put in cleaner burning stoves with chimneys, for example, you don't really reduce the incidence, but you can reduce the severity of the respiratory infections. And I don't want to talk all about the hazardous air pollution and the indoor stuff, but we really now know that you can't burn biofuels and get low enough air concentrations of particulate matter to avoid adverse health effects. So anyhow, there are a bunch of studies that are coming up that now document the modulation of the innate pulmonary system response for inflammation, alteration in macrophage function, and upregulation of viral and bacterial receptors. So another example of an infectious agent environmental interaction going on is the whole story with Hep B and cancer and aflatoxin. I think, and the data here on the slide just shows you that if you have Hep B, you have an increased risk of hepatocellular carcinoma. Aflatoxins by themselves also have a small increased risk. When you put them together, you get a tremendous synergy. So again, the interaction between the infectious agent and the toxicant. And I think it's important, this is just showing, we think of aflatoxin on corn, but we should be thinking of it on a lot of other different kinds of grains and nuts and so on. And as climate is changing, we are going to see that mold growth is going to be more and more of a problem. So a new interaction that we're currently looking at is the whole issue of Zika. I think the world was taken by surprise with the anencephaly that was occurring, especially first in northeastern Brazil in 2015 and 16. There have been other babies who have been infected by Zika. Most of them were infected in Brazil. The ground zero for Zika infection in the US was not Miami, which you heard lots about, but was in Puerto Rico, where there were over 50,000 cases documented in 2016. But then there's almost been nothing since. And the question is, has the virus really gone away? Or have most people had infections that basically didn't result in any adverse effects because they were able to handle it? And one of the hypotheses is that the reason for the severity of the brain effects of Zika in Brazil may have been related to co-exposure to certain environmental pesticides that were in use there. And the one that people were very interested in was pyriproxyfen, which is a larvicide, which was used in drinking water. to do away with places where you had standing water. And there have been 2 studies that have done so far. One is ecological and one is a case control. They didn't see any evidence for an interaction. The National Toxicology Program has been conducting some standard developmental and reproductive toxicity studies in rats and rabbits. They're not seeing any effect of pyriproxyfen. They are also doing some studies in zebrafish. There's a suggestion of effect on the developing brain in those studies. But the point is the NTP is not looking at an interaction between the pyriproxyfen and the Zika virus. That would be the next step to look at. Jose Cordero and company are studying a Puerto Rican cohort under our Superfund program and under our environmental health core centers. We have 2 cohorts in Puerto Rico that are linked, the PROTECT and the CRECHE cohorts. One is a birth cohort, and then when the children get to the age of 1, they continue to be followed in the other. And as part of the Zika in pregnancy study, which is a cross-NIH effort that involves NIAID and Child Health and us, we are following over 1,000 pregnancies where there was Zika infection and looking for any adverse health impacts. It's been hard because basically the episode is kind of past, but we're still continuing to look at that. I did want to make a quick point about metals and endocrine disruptors and pesticides and their role altering the microbiome, again, the bacteria in and on us. And so there are studies mainly in mice, which are showing alterations in the microbiome and associated metabolites from multiple exposures. We're going to hear, I think, some more, maybe about arsenic later in this from Margaret Karagas. There's data with lead, manganese, obviously PCBs still continue to be of interest, and diazinon, which is a major pesticide. And some suggest that the metabolic changes could interact with obesity, atherosclerosis, and neurologic diseases. So the graphics are just some of the information showing the increased risk when you have the interaction going on. I mentioned climate change before, and I think anybody who's a gardener, when you went to buy your seeds a couple of years ago, you found that the range for the time of year, your part of the country had changed, and all of a sudden you could plant your crops sooner because the last frost day was earlier, for example. But for example, if you look at expanded the range of some vector-borne diseases, what you can see here, the one on the right is looking at a comparison of the range of dengue in Nepal, looking at two different time points. Let's see, put on my glasses, 2030 and 2070, and comparing that. And there's the area of transmission, and it's increasing and moving further north. But I think the example of Lyme disease in the northern U.S. and southern Canada is especially important. And when you look at this, what you can see is that up until 2000, Lyme disease was primarily, in the northern tier of the U.S. and a little bit of Canada. But look what's happening. The ranges are expanding into more into further north into Canada, further north into our Great Lakes regions and so on. And with the modeling, first of all, we know that has happened now in 2019, but also with the predictions that are going on from the different climate change modeling. So I think the important conclusions is that environmental pollutants affects how we are infected, and I should say how we are infected is going to affect our response. Understanding environmental immunotoxicity will be critical to making successful vaccines, and the scientific frontiers going forward include microbiome interactions and effects of global environmental change. So thank you.